You probably think, you know what a vaccine is. I mean, most of us do, right?
Right, yeah. You'd think it's pretty straightforward.
Yeah, but over the last few years, the actual dictionary definition of that word has been, well, it's been fundamentally and kind of controversially shattered.
Completely.
If you look at the technical language used in the world of immunology today, we're navigating this diagnostic and semantic landscape that is basically shifting right under our feet.
It really is.
And for you listening, this isn't just, a debate for linguists. This shifting definition has been at the center of a massive public
Yeah,
discourse.
because we're witnessing a scientific paradigm shift playing out in real time.
Hmm.
The way medical science understands how to interact with the human body has evolved so dramatically that, honestly, our old vocabulary simply couldn't contain it anymore.
Which is exactly our mission for today. We are taking a deep dive into an incredible stack of pharmacological material all about vaccines and immunizing agents.
Fascinating stuff.
It really is. We're going to explore how these medical marvels actually train your immune system, the wild history behind them, the brilliant ways we've engineered delivery mechanisms into the body, and here's where it gets really interesting. Why relying on quote-unquote natural immunity is sometimes the absolute worst way to protect yourself.
I'm so looking forward to jumping in. We can, break down the biology and the pharmacology behind these concepts because understanding how your immune system gets programmed, it isn't just for medical students anymore.
No, not at all. It is essential civic literacy at this point. So let's start with the absolute basics, because the material points out a big umbrella term that causes, I think, a lot of confusion, which is immunizing agents.
Right.
And underneath that umbrella, we have two very different approaches, active and passive. So how do we pull those apart?
Well, it really comes down to who is doing the biological work.
Okay.
So active immunizing agents, which are your vaccines and toxoids, they prompt your own immune system to build its own lasting defenses.
So your body does the work.
Exactly. They force your body to manufacture memory cells.
Oh.
But passive immunizing agents, on the other hand, are things like immunoglobulins or antitoxins. Those are borrowed
Borrowed? Like from where?
from a lab, usually, or from someone else's immune system. They are pre-made antibodies that are just injected directly into you.
Oh, I see. So active immunity is essentially teaching your immune system to fish.
Yes.
While passive immunity is like someone just handing your body a fish for dinner when it's in a state of emergency.
That is a brilliant way to look at it. Yeah, because when someone hands you a fish, you eat it, and then it's gone.
Right.
So if you step on a rusty nail and go to the ER, they might give you a tetanus immunoglobulin.
Just to be safe.
Right, and that is a rapid deployment of borrowed antibodies to immediately neutralize any toxin floating in your blood. But proteins break down.
So it doesn't last.
Exactly. And because your body didn't make them, it has no idea how to make more.
Oh, wow. Okay, so that's why they also give you a tetanus toxoid vaccine at the same time.
Precisely. That's the active part. That teaches your own cellular factories to recognize the threat so you are protected for the next decade.
That makes total sense.
And you see this passive approach happening right now with the new RSV monoclonal antibodies given to infants.
Oh, right. I've read about that.
Yeah, it provides this immediate temporary armor while their own immune systems are still, maturing and figuring things out.
Okay, so teaching the body to fish. To understand how we originally figured that out, we have to talk about the origin story, which involves, and this is wild, literal cows.
It really does.
We are going back to 1796 with the English physician Edward Jenner. He noticed that local milkmaids, who frequently caught a mild disease from cattle called cowpox, seemed to be, miraculously immune to smallpox.
Which was a huge deal because smallpox was historically a global terror. I mean, it was killing something like 30% of the people it infected.
30%? That's devastating.
Yeah, it really was. So Jenner decided to test this folk observation. He took matter from a cowpox sore on a milkmaid named Sarah Nelms, and he actually scratched it into the arm of an 8-year-old boy named James Phipps.
Which sounds completely unhinged by modern standards.
Oh, absolutely. You could not get ethical approval for that today. And then weeks later, he deliberately exposed the boy to actual smallpox.
And he didn't get sick.
James Phipps didn't get sick. Now, Jenner didn't invent the concept of inoculation. People had been using actual smallpox material in a really risky practice called variolation for centuries.
Oh, right, where they would inhale scabs and
Yeah.
stuff.
But Jenner popularized a safer substitute using a related, milder animal virus.
But he didn't call it a vaccine, right? Because that word didn't even exist yet.
No, Jenner just called it cowpox inoculation.
Hmm.
The term vaccine didn't actually show up until roughly 80 years later.
Wow, that long.
Yeah, the French chemist Louis Pasteur coined it in the 1880s, and he derived it from the Latin word vacca, which means cow.
Which is pretty funny when you think about it, considering Pasteur's own breakthrough vaccines for things like anthrax and rabies had absolutely nothing to do with cows.
Right. He did it explicitly as a tribute to Jenner. Pasteur's 1885 rabies vaccine was actually created by drying infected rabbit spinal cords to weaken the virus.
Which is pretty wild pharmacology for the 1880s.
It really was, and that was the direct biological forerunner to the modern live attenuated vaccines we use today.
And for most of the 20th century, that was the standard, right? The word vaccine inherently meant taking a whole physical organism, a virus or a bacteria, weakening it or killing it, and putting it into a human body.
Exactly. It was a physical thing.
But looking at the modern pharmacology, this is where the dictionary definition starts to crack. We see a major shift in 1986, and then the whole paradigm basically shatters in 2020.
Yeah, the literal language had to evolve because our engineering evolved. So in 1986, the first recombinant subunit vaccine was licensed for hepatitis B.
And that wasn't a whole organism.
Not at all. It was just a single viral protein that scientists managed to grow in yeast cells. There was no bug in the vial at all, just a manufactured piece of one.
And then came 2020 and the mRNA COVID-19 vaccines. I really want to pause and dig into this because this specific transition is where the public debate just exploded.
It certainly did.
Between 2021 and 2023, public health agencies actually went in and revised their official glossary definitions to accommodate mRNA technology.
Right.
And critics looked at this and argued that authorities were basically moving the goalposts just to classify this new technology as a vaccine, while, immunologists countered that they were simply keeping up with advanced biology.
It was a very heated semantic debate.
Right, so impartially speaking, if we aren't putting the actual bug in the vial anymore, what exactly are we giving people? Like, how does injecting pure information protect me from a physical virus?
Well, it's all about biological first principles. To your immune system, the essence of a pathogen isn't the whole bug. The essence is the antigen.
The antigen.
Right. An antigen is just a specific molecular structure, a 3D shape on the outside of the virus that your adaptive immune system can actually recognize.
Okay, just the shape.
Exactly. The entire goal of any immunization is simply to show your immune system that specific shape in a controlled environment so it can build memory B cells and memory T cells.
So when the real dangerous disease finally shows up, your immune system already has the blueprints on file.
Right. And it can mount a massive defense before the virus can replicate out of control.
That makes sense.
And historically, the only way we knew how to deliver that shape was to deliver the whole weakened bug. Then, in 1986, we figured out how to manufacture and deliver just the isolated shape itself.
The Hep B vaccine you mentioned.
Right. And with mRNA, we figured out how to deliver the genetic instructions for that shape.
So we aren't even delivering the shape anymore.
No, the mRNA enters your muscle cells and essentially tells your own cellular machinery to temporarily manufacture the shape, the viral protein internally.
Oh, wow.
But the end result for your immune system is identical.
Mm-hmm.
It sees the antigen, and it builds memory cells.
So the definition had to change because our delivery mechanism went from shipping fully assembled cars to shipping isolated car parts to finally just emailing the CAD file so your body cells can 3D print the part itself.
That is exactly what happened, yes.
Going from literal cows to microscopic CAD files, that is an incredible progression. So since modern immunology is really just an information delivery problem, let's explore how we actually solve it. The material breaks down 5 core delivery mechanisms. Let's walk through the biological logic here.
Sure. So the first approach is the oldest, which we just touched on, live attenuated vaccines.
Right.
This includes the MMRSO, measles, mumps, rubella, the oral polio vaccine, yellow fever, and the chickenpox vaccine. You are using a weakened but still living version of the pathogen.
And because it's alive, it actually replicates a little bit in your body, right?
It does. It replicates slowly.
And because it mimics a natural infection so closely, it produces this massive immune response. I imagine that's why these often give you durable, lifelong immunity from just a single dose.
They are incredibly effective for exactly that reason. But there is an inherent trade-off. Because the organism is alive and replicating, it generally cannot be given to people with severely weakened immune systems or to pregnant women.
Because even a weakened bug could theoretically overcome a compromised system and cause disease.
Exactly. It's too risky.
So if a live bug is too risky for vulnerable people, the logical next step is to just kill the bug entirely, right?
Which is the second mechanism, inactivated vaccines.
Okay.
Here, the pathogen has been completely destroyed using chemicals or heat, so it cannot replicate at all. Jonas Salk's famous injectable polio vaccine is the classic example, along with hepatitis A and most seasonal flu shots.
So they are much safer.
Yes.
But I'm guessing because the bug is dead and can't replicate, your immune system doesn't get quite as riled up about it.
That is the downside. Your immune system clears the dead pathogen relatively quickly, which means the cellular memory fades faster. And that is why inactivated vaccines almost always require multiple booster shots to maintain protection over time.
Okay, so if we want to get even safer than a whole dead bug, we move to the third mechanism. Subunit, recombinant, and toxoid vaccines. This is the 1986 shift you mentioned earlier.
Right. We just take a tiny fragment of the pathogen, or a deactivated toxin. The hepatitis B, HPV, and the diphtheria and tetanus vaccines fall in here.
Now, because these are just isolated proteins, they are incredibly safe. But I'm a little confused by the pharmacology here.
Okay.
I read that these often require something called an adjuvant like an aluminum salt to actually work. If you're already handing the immune system the protein shape, why does it need extra chemicals?
Well, because an isolated protein floating by is actually pretty boring to your body.
Boring.
Yeah. Your immune system has two main branches. The innate system, which acts as a general alarm, and the adaptive system, which builds the specific memory cells we want. The adaptive system won't bother building memory cells unless the innate system sounds the alarm first.
I see.
A pure, clean protein doesn't always trigger those alarms. So the adjuvant provides a localized biochemical danger signal, a bit of irritation or inflammation, that forces the innate immune system to wake up and pay attention to the protein.
So the adjuvant is essentially a built-in alarm system.
Yes.
It's there to shout, hey, look over here, this is a threat. Because the protein on its own is wearing camouflage.
That's a great way to think about it. And speaking of camouflage, that actually brings us to a major biological hurdle and our fourth delivery mechanism, which is conjugate vaccines.
Okay, this part of the material was fascinating. It highlights a brilliant solution to a very specific problem with babies, where the bacteria is basically wearing an invisibility cloak.
Yes, it's a huge problem. Certain highly dangerous bacteria, like Haemophilus influenza type B, or Hibs, and pneumococcus, disguise themselves in a plain, slippery sugar coating called a polysaccharide. The problem is, an infant's developing immune system is notoriously bad at recognizing and responding to plain sugars. Their B cells just ignore it.
So the bacteria can slip right past their defenses.
Exactly.
So how do you train an infant's immune system to attack something it fundamentally can't see?
You trick it. Scientists figured out how to chemically link or conjugate those invisible sugars to a carrier protein that the infant's immune system does recognize aggressively.
Like what?
Like a harmless deactivated tetanus toxin. It's a Trojan horse in reverse.
Oh, wow.
When the infant's immune system attacks the highly visible carrier protein, it is forced to simultaneously process the sugar coating attached to it. It basically trains their immune system to recognize the disguise.
That is unbelievably clever.
Isn't it?
And the results are staggering. The material notes that the Hib conjugate vaccine, when it was introduced in the late 1980s, cut infant Hib meningitis by over 90%.
It was incredible.
It's a massive medical triumph that almost nobody talks about today, simply because the disease vanished before today's new parents were even born.
It is the ultimate paradox of public health. When the engineering works perfectly, nothing happens.
Right. Which brings us to the modern era, where growing whole bugs or tricky proteins in vats of yeast is too slow. The fifth mechanism, genetic vaccines. This includes viral vectors and mRNA.
Right.
We are skipping the physical delivery of the antigen entirely and just sending the instructions. But how do these two differ in how they actually get the code into our cells?
Well, viral vector vaccines, like the Johnson & Johnson or AstraZeneca COVID vaccines and the Ebola vaccine, use a harmless, unrelated virus, usually an adenovirus, as a delivery truck.
Okay.
They basically hollow out the adenovirus and put the DNA instructions for the target antigen inside. The adenovirus injects that DNA into your cell's nucleus, which then creates the mRNA to build a protein.
And the mRNA vaccines like Pfizer and Moderna, they skip the viral delivery truck completely.
Exactly. They just package the fragile mRNA instructions directly in a microscopic bubble of fat, a lipid nanoparticle. That fat bubble naturally fuses with your cell membrane and drops the instructions directly into the cellular fluid, completely bypassing the nucleus entirely.
Wow.
And once you have this lipid nanoparticle platform, swapping out the target disease is purely a matter of typing a new genetic sequence into a computer.
Which explains the unthinkable speed. Moderna went from receiving the SARS-CoV-2 genetic sequence to shipping vials for a phase 1 human trial in about 63 days.
63 days, yes.
Historically, that process would take a decade.
Easily a decade. It is essentially plug-and-play pharmacology.
It's incredible. But look, whenever medical science starts manipulating biology at this microscopic level, people naturally get nervous.
Of course.
We need to address the real-world interactions and the widespread fears about how these mechanisms impact our bodies. A huge one is the fear that the modern childhood schedule is simply overloading an infant's immune system.
It is a very common anxiety for parents, and it's understandable. But if you look at the immunology, the math simply doesn't support the fear.
Really? Why not?
Because to an immune system, an antigen isn't viewed as one big object. It is broken down into tiny, recognizable surface features called epitopes.
Okay.
If you add up every single epitope in the entire modern childhood vaccine schedule, it is a vanishingly small fraction of the immunological data and infant processes every single day.
Just from crawling on the floor, putting dirty toys in their mouth, their own gut microbiome.
Precisely. Immunologists have actually calculated that, theoretically, an infant's immune system has the capacity to respond to thousands of vaccines simultaneously without being exhausted.
Thousands. At once.
At once, your immune system is a parallel processing supercomputer. A scheduled immunization is barely a rounding error in its daily workload.
Okay, let's tackle another major myth. You hear this all the time. Natural infection is always better. Let them get the chicken pox. Let them get measles. It builds a stronger natural immune system.
Right.
But wait, isn't that premise actually incredibly dangerous? I read in these notes about a 2015 study published in Science by Mina and colleagues that completely upends this idea.
It was a landmark study, yeah, and it revealed something terrifying about a natural measles infection. Surviving measles causes something called immune amnesia.
Immune amnesia. That sounds like your immune system is getting hit with a malware attack that literally wipes the hard drive of all your previous defenses.
That is biologically exactly what is happening.
Really?
Yeah. The measles virus doesn't just make you sick, it specifically infects and destroys your memory B cells and memory T cells.
Oh my God.
So even if you successfully survive the measles, your immune system has physically lost the cellular memory of how to fight off all the other bugs you've encountered in your entire life.
Wow.
Survivors are left highly vulnerable to completely unrelated infections for up to 2 or 3 years afterward. The vaccine provides the measles immunity without wiping your hard drive.
That completely dismantles the natural is better argument.
Completely.
And it's not just measles. A natural chickenpox infection leaves the live varicella virus dormant in your nerve tissue for the rest of your life.
Yes, it does.
And decades later, when your immune system naturally weakens with age, it can reactivate as shingles, which is excruciatingly painful. The attenuated vaccine significantly lowers that long-term risk because the weakened strain is much less likely to reactivate.
Exactly. It's all about risk mitigation, not just for the individual, but at a population level, which brings in the concept of herd immunity.
Right, herd immunity.
When a high enough percentage of a population is immune, chains of transmission are broken. The virus simply runs out of susceptible bodies to jump to, which indirectly protects the vulnerable people who can't get vaccinated, like newborns or cancer patients.
And the math behind herd immunity relies on something called the basic reproduction number, or R0.
Right. R0 is a measure of how insanely contagious a disease is.
Okay.
Measles has an R0 of 12 to 18.
Wow.
That means one infected person in a totally susceptible population will, on average, infect 12 to 18 other people.
That's incredibly fast spread.
Because it spreads so aggressively, you need roughly 95% of the population to be immune just to prevent sustained outbreaks. If vaccination coverage drops to 90 or 92%, measles will immediately find the cracks.
Which means public trust is the absolute glue holding this entire public health system together.
It is the only thing holding it together.
And when we look at the high stakes of maintaining that trust, we see tremendous triumphs, but we also see tragic scandals. Let's look at the ultimate triumph first. Smallpox.
Smallpox remains the ultimate proof of concept for herd immunity. The World Health Organization launched an intensified global eradication campaign in 1967.
And it worked?
A decade later, in 1977, a hospital cook in Somalia named Ali Malmolen was recorded as the last known case of naturally occurring smallpox.
And by 1980, the WHO formally declared the disease eradicated from the planet.
It's the only human disease we have ever completely wiped out.
It's amazing. But the material also highlights the dark chapters, and the contrast between two specific historical events really struck me. First, there's the 1955 Qatar incident.
Right, so when Jonas Salk's inactivated polio vaccine was finally approved, several companies rushed to manufacture it to meet massive demand.
And one of them messed up.
Qatar Laboratories had a severe flaw in their production process. The virus in their batches was not fully killed by the formaldehyde. They accidentally shipped lots of vaccine that contained live, virulent, paralyzing polio.
The fallout was devastating. I mean, 200,000 children were exposed.
Yes.
It caused 40,000 cases of abortive polio, 56 cases of severe paralysis, and several deaths.
It was a catastrophic manufacturing failure. But crucially, the immediate government response to that disaster led directly to the creation of the intense, strict FDA vaccine manufacturing oversight and safety regulations that we rely on today.
Okay. And then you contrast that genuine physical disaster with the 1998 Wakefield affair.
Right.
Andrew Wakefield publishes a study in The Lancet claiming to link the MMR vaccine to autism based on a sample size of just 12 children. The study was totally fraudulent. He completely falsified the data, and he had massive undisclosed financial conflicts. He was literally being funded by lawyers who were actively suing vaccine manufacturers.
It's staggering.
It was eventually fully retracted, and he lost his medical license.
But the irony is profound and deeply unsettling. Qatar caused acute, tragic physical harm, but birthed systemic safety and rigorous oversight.
Right.
Wakefield caused no direct physical harm with his paper, but birthed a systemic hesitance that brought measles roaring back globally. It shows just how fragile public trust and herd immunity really are when confronted with fabricated misinformation.
It's completely fascinating and honestly a bit terrifying. And to be fair, it's not always human error or fraud that complicates things. The biology itself can create tricky situations. I want to ask about something called circulating vaccine-derived poliovirus, or CVD-PV.
Okay.
How does a vaccine actually cause a virus to circulate?
This is a rare but very real biological complication. So remember, the oral polio vaccine uses a live, attenuated virus because it's cheap, easy to administer, and creates strong gut immunity.
Right.
It replicates in the intestines and is actually shed in the person's stool. In areas with incredibly low vaccination rates and poor sanitation, that weakened virus can actually circulate from person to person through contaminated water.
Oh, wow.
And if it circulates through a vulnerable population long enough, the virus can continuously mutate until it reverts back into a form capable of causing paralysis.
So the vaccine's own living biology creates a secondary risk.
Exactly. Which is why wealthier countries switch strictly to the injectable killed version once wild polio is eliminated from their borders.
That makes total sense.
And because there are rare but real biological risks, whether it's CVD-PV in developing nations or the extremely rare severe allergic reactions to modern vaccines, the U.S. government created the National Vaccine Injury Compensation Program in 1986.
To handle those rare cases.
Right? It acts as a systemic backstop to handle rare injuries fairly, ensuring manufacturers keep making vaccines and basically keeping the whole public health effort stable.
Which brings us to the core message for you listening today. Vaccination is arguably the highest leverage tool in the history of modern medicine.
Oh, absolutely.
It has driven historic global declines in child mortality. Yet the WHO now lists vaccine hesitancy as one of the top 10 threats to global health.
It's a real crisis.
Understanding this pharmacology, knowing what an antigen is, how your cells process information, what a vaccine actually does and doesn't do, it is no longer just a biology lesson for a textbook.
Yep.
It is essential civic and scientific literacy in the modern world.
It truly is. We are interacting with microscopic information systems every single day. And, well, before we wrap up, consider this. We discussed earlier how a seemingly mild natural infection like chickenpox actually hides out dormant in your nerve tissue for your entire life, waiting to strike back decades later as painful shingles.
Right.
It really makes you wonder, what other long-term hidden cellular damage or dormant time bombs might be left behind in our bodies by other so-called natural infections that medical science simply hasn't discovered yet?
Wow. If doing things the natural way leaves undetected time bombs in our nervous system, suddenly a little lab-made mRNA instruction manual doesn't seem so intimidating after all.
Exactly.
So going back to where we started, the dictionary definition of a vaccine might have changed. It might have shifted from delivering a physical pathogen to delivering pure genetic information, but the goal remains exactly the same, building an immunological bridge to a safer future.
Well said.
Thank you for joining us on this deep dive. Keep questioning, keep learning, and we'll catch you next time.